Harshad, we're here at last.
My name is Harshad Vishwasrao. I'm a postdoc in Eric Kandel's lab. One of the most important questions in neurobiology today is how neurons in the human brain interact with each other to produce these phenomena called learning and memory, thought and consciousness. But you run into a problem when you try to do experiments, which is that the human brain is incredibly complex. It's on the order of about 100 billion cells in the human brain, 100 billion neurons in the human brain. And what that means is that doing meaningful experiments becomes incredibly difficult. So a while back, neuroscience developed a strategy to approach this problem, which is to work on simpler animals with simpler nervous systems and that have simpler behaviors. Well, one good example of such an animal is the sea snail, Aplysia. Aplysia has a very simple nervous system. There's only about 20,000 neurons. And these neurons are very easy to identify. It also has very simple behaviors. Now, the behavior that we've been focusing on for a long time is something called the gill withdrawal reflex. You poke the animal in just the right place and its gill withdraws, pulls in very rapidly. It's a protective response. Now, it turns out that the circuit that controls the gill withdrawal reflex is very simple. It consists of only a handful of neurons. There's a particular synapse that's involved in the gill withdrawal reflex, a synapse between a sensory neuron and a motor neuron. And these are very specific cells. Now, the strength of that synapse controls the strength of the gill withdrawal reflex. So we identify those neurons, we pull them out of the animal, and we can put them in a dish with a solution that mimics the body fluid, and they will grow and they will form the same synapse in the dish that they form in the animal. For example, here we've got a sensory neuron emitting multiple axons, and this one goes and forms synapses with this motor neuron, and this axon comes over here and forms synapses with this motor neuron. Now I can take a sensory neuron and put it in a dish all by itself. And that sensory neuron will start sending neurites out in all directions, constantly growing, constantly looking for its postsynaptic target, that motor neuron. Now, we haven't provided that target here, but the sensory neuron will continue growing until it finds that postsynaptic target. Now, each one of these growth events in and of itself is a very complicated mechanism. In fact, if we focus in on a single growing tip, we find that there's an immense amount of molecular structure, molecular dynamics that's going on. Here, for example, we're looking at one growing tip, and the little glowing dots that you see are granules of actin RNA. RNA is basically an instruction set for how to make the actin protein, which is a very important molecule taking part in growth, and it generally forms the skeleton of the cell. Now, when we also provide the postsynaptic partner to the sensory neuron, that is, if we put the motor neuron right next to the sensory neuron, then the sensory neuron stops growing in all directions, and it sends its processes specifically to that motor neuron. So what you're seeing here are sensory neurites in green and motor neurites in red. And you find that the sensory neurites grow along the motor neuron. And in particular, you start to notice small swellings in the sensory neurites. These are called varicosities. That's where the synapses actually form. That's where electrochemical communication occurs between the two cells. So each one of those small swellings or varicosities in turn contains quite a bit of structure. So what we're looking at here is a single varicosity rendered in 3 dimensions. And what you find is that the varicosity is in green, that's the presynaptic neuron, and it's synapsing on neurites of the postsynaptic neuron, which are in red. The blebs that you see, the small spheres, the little green spheres, contain a molecule called synaptofluorin. That's actually specific to synaptic vesicles, so it's generally used as a marker for synapses. So once a synapse between a sensory neuron and a motor neuron is formed, we can actually modulate its strength in the same way it's modulated in the intact animal. As we expose the synapse to a specific treatment of serotonin. Serotonin is a chemical that increases the strength of that synapse, and the animal is responsible for increasing the strength of that gill withdrawal reflex. So for example, here we've got a synapse that's formed between the sensory neuron, appearing in yellow, and the motor neuron appearing in red. And we can treat the cells with serotonin, and if we come back 24 hours later, we find an enormous increase in the amount of contact between the two cells. The synapse in this case has become greater. There are more varicosities, there are more synaptic contacts. Essentially, when you see an increase in the strength of a synapse, that's learning.